My great uncle was a high school math teacher, and he taught me 3 extremely valuable lessons: how to ride a bike, how to tell a story, and how to do geometry. However, as he grew older and was diagnosed with Alzheimer's, he forgot these very lessons that he passed down to me. Alzheimer's is a neurodegenerative disorder that slowly erodes memory and cognitive function. For me, once I joined college, I knew after seeing my great uncle's story that I wanted to study this deeply troubling disease. And so as a result, I joined the Dr. Lipkova's Octopath lab, which is a lab that uses AI to study the pathological progression of diseases. The project that I'm working on is in collaboration with Dr. Manuki's choroid plexus epithelial lab, where they study how the choroid plexus functions. So the choroid plexus is a region of the brain that provides cushioning against impact. When looking at these cells underneath a microscope, researchers found a rare type of cell known as oncocytes. Oncocytes are cells that are swollen with mitochondria, and these researchers at the Manuki lab hypothesized that they may be correlated with the progression of Alzheimer's. However, the issue is that these cells are extremely rare, and we can see this in the image on the right, where we are looking at a whole slide image of 100 cells, and from here, only 3 of these hundreds of cells were labeled as oncocytes. And so as a result, researchers at the Manuki lab spend hours upon hours painstakingly and manually labeling and annotating these cells. So that's where AI comes into play. Right now, what I'm doing is I'm taking an existing deep learning model that is known as the HoverNet model and adapting it specifically to detect oncocytes. The idea is to take these labeled images that the Manuki lab has already done and fine-tune the model so that it can specifically segment cells and then classify them as oncocytic or not. And so far, the early results are pretty promising. What we've seen is that across different tissue types, the model does a great job of not only identifying cells, but also determining what their cell type is. And that's seen in these images where each color represents a different cell type. So for me, what I'm going to do next is take this model and have it trained specifically on choroid plexus epithelial tissue cells, and eventually, hopefully, release this tool so that it's an open source tool that researchers and clinicians worldwide can use to ideally speed up how fast we treat Alzheimer's. And for me, this project is deeply personal. My Alzheimer's stole a lot of memories and valuable time from my great uncle, and I hope that through this project I can help preserve memories, protect families, and speed up the fight against Alzheimer's. Thank you so much.